Let me check my notes, Veronica. I just saw your thing. Let me check my notes. Maybe I'm remembering it wrong. Uh, yeah, that would be a typo. I need to fix that. The ciliary muscle is not also called the ciliary body. It's a part of the ciliary body, but it's not also called that. Okay, sorry about that. So the answer should be option B for 5. Okay, so for question 6, the answer to question 6 is D. So the lens focuses objects onto the retina, right? The retina is that area that is having all those rods and cones, and the lens functions to focus objects onto it. So if you answer D, that would be the correct answer. Cornea, the cornea right here, this is where light comes in. So this is a see-through part at the front of the eye. So it's not needing objects to be focused onto it. The cornea kind of like lets light in, if that makes sense. The lens is held in place by ligaments, but they're called suspensory ligaments, not denticulate ligaments. So that's why that's right. Denticulate ligaments are the ligaments that hold the spinal cord in place. They're an extension of pia mater that holds the spinal cord in place. Okay. Question number 7. The area of increased surface area to accommodate more rods and cones is which of these options? Good. A, right? So that's the definition of macula lutea. Fovea centralis is the area of most acute vision. Okay, so the area that accommodates more rods and cones, macula lutea. The area of most acute vision, fovea centralis. Okay, question 8. The area in the eye from the cornea to the iris is the it's the anterior chamber. Okay, so the anterior chamber goes from the cornea to the iris. The posterior chamber goes from the iris to the lens, and the anterior cavity goes from the cornea to the lens. And the posterior cavity would basically be from the lens back. So if you're thinking about the eye, that would all be posterior that I just drew. I'm kind of drawing this big so we can all see it. So this is my lens right here, right? And then here's my iris out here. So if I'm drawing it, this section here would be the anterior chamber. It goes from the cornea on the outside to the iris, which is right here. This is the iris. Now from the iris to the lens, here, do a different color. Here, this would be the posterior cavity, okay, posterior chamber. And then the anterior cavity is both of those. So the anterior cavity is just the reference for that whole section would all be anterior cavity. That kind of makes sense. So the anterior chamber is the front half, posterior chamber is the back half of the anterior cavity, versus posterior cavity is all of the rest of the entire eye. So for number 8, the answer is A, because that's the definition. From the cornea to the iris, halfway back, that's where we have the anterior chamber. Okay? Kind of just a memorization thing on that, but hopefully the drawing helped a little bit. Question number 9. When aqueous humor builds up, the condition is called... Good. It's called glaucoma, right? Glaucoma is when we get excessive aqueous humor, which is like the jelly-like stuff in your eye, built up and not reabsorbed. So it's kind of like it's collecting inside of your eye. Anopsia is without normal vision. So someone who has some type of impediment in their vision would have anopsia. Myopsia is being nearsighted. Hyperopsia is being far-sighted. So maybe you could just think about myself. I am near to myself. I'm nearsighted, near to myself. Myopsia, I'm near to myself. Hyper is far-sighted. Maybe you could just think of hyperactive, you travel far. Maybe you could think of that. So glaucoma is the only one that matches the definition of the question, but you have your definitions for A, B, and C as well. All right, question number 10. The middle ear, which does not belong for the middle ear? Good. So E is the correct answer. The malleus, incus, and stapes are the bones of the middle ear. And then the eustachian tube or pharyngotympanic tube equalizes pressure between the outside air and the inside air. And that's also part of the middle ear. So the answer here would be none. Question 11, the semicircular canals, which is not true. So remember, our semicircular canals, they kind of look like this. They do have, the entire thing is called the ampulla. Inside the ampulla, there's another structure called the cupula. Cupula is like an additional area of cells to increase surface area. The stereocilia are the ones that depolarize in the semicircular canals. Okay, so the stereocilia are the ones that are attached to the dendrite. So C, stereocilia are attached to the dendrite. That would be true. It does have the ampulla on the outside, and then the cupula is this part right here, and then the stereocilia are attached to the dendrite. So ampulla is the rounded portion, cupula is the part inside. stereocilia are attached to the dendrite. And then we see that there are two types of fluid. There is the perilymph, which is a thick fluid on the outside. So D is going to be the correct answer. So here's my perilymph. And then my endolymph is my thin fluid on the inside. So this right here would be my, oops, this right here would be my endolymph. Okay, so that's the thin fluid on the inside. So your answer to this question would be D, because we should have endolymph as the thin fluid on the inside. Good? All right. And as always, if you guys have questions on something or it's not clear, please ask in the chat. I'm not going to pause and ask if you have questions all the time, but if you have any questions, please ask, and then I will be happy to answer those. I just don't want you guys to, feel like I'm always asking if you have questions, but if you have questions, please, I want to hear them ask in the chat. Okay, question 12. The utricle, what does it do? What does the utricle do? One's actually a little bit of a tricky one. All right, so remember the utricle and the saccule are both a part of the vestibule or the otolith organ. So the vestibule or the otolith organ is the organ that creates rapid movement or roller coaster sensation. So that would be the function, is to create rapid movement or roller coaster sensation. It's going to perform that using the utricle. The utricle is for back and forth motion. The saccule is for up and down. The way you can remember that is like you would drop a sack, drop a sack of potatoes, drop a sack down. That's how I remember it. So saccule is for up and down. You would drop a sack down, versus utricle is back and forth. So we know that the answer is not C. The utricle and saccule are different things. The saccule is the one performing up and down, so it's not A. It is not located in the semicircular canals. It's located in the vestibule, but it does function for rapid movement sensation, right? The utricle and the saccule together create rapid movement sensation. So the answer to 12 would be D. Does that make sense? Kind of eliminate all the other choices. So our answer is D. Good. Any questions? All right. Question 13. Which of these has the dendrites in the cochlea? So our scala vestibuli and scala tympani, these are both perilymph filled outside layers to the cochlea. And the scala media is also called the spiral organ of Corti or the cochlear duct. And the scala media is where we actually have the endolymph and hearing. So hearing is generated in the scala media. It kind of looks like this. We have our basilar, our tectorial membrane, our basilar membrane, and then we have these stereocilia cells, stereocilia in the certain cells that are embedded in the tectorial membrane in order to allow continuous sound. So this is the tectorial membrane. This is the basilar membrane. Together, these make up the scala media. I'm trying to kind of draw these so you guys have a quick refresh of this info. And then these would be the stereocilia. Those are the ones attached to the dendrite. Okay? So our little recap here. This one with the kinocilium, the stereocilia, and the calcium carbonate crystals, right? This one. That is the utricle and the saccule for the vestibule. Okay, so this is the utricle and the saccule. That would be our kinocilium, is our dendrite. Then we have our, this one I just drew, the ampulla, the cupula, right? It has the perilymph and the endolymph, and it has the little stereocilia. Okay, so this would be the cupula with the stereocilia, and this is part of the semicircular canal. Remember, semicircular canal is for balance. Vestibule is for rapid movement. And then our third sensation over here is the cochlea. It's for hearing. And we have the stereocilia again. Okay, so our dendrite, highlighting in red, dendrite for hearing, stereocilia. Dendrite for rapid movement, kinocilium. Dendrite for the semicircular canal, balance, stereocilia. Good? So hopefully that's a good little visual all on one screen for you guys to see the dendrites for each of those sensations in the ear. Remember, you also have taste, which is a chemoreceptor, right? Chemoreceptor for taste, and that is the olfactory cell. Then you have the chemoreceptor for smell, and those are the cells in the cribriform plate. And then you have your eye, which is the rods and the cones, and those are photoreceptors. Okay? So the ear is probably the trickiest one because they're all kind of start to blend together. So hopefully that helps seeing them all on one screen. Okay, any questions on the ear? We're moving on to kidney. All right, so question 14. The glomerulus, which of these is not true about the glomerulus? Good. D, right? Because the vasa recta are the blood vessels that go around the loop of Henle. So if that's the case, they would not be part of the glomerulus. Okay, so again, vasa recta are the blood vessels that go around the loop of Henle, so they're not part of the glomerulus. Question 15. There are how many types of nephron? two, right? So there are two types of nephron. There's the cortico and the juxtamedullary nephron. Those are the two types, cortico and juxtamedullary. So remember, the corticonephron is mostly in the cortex, and the juxtamedullary nephron is mostly in the medulla, but both have their loop of Henle in the medulla, but those are the two types of nephron. All right, question 16. The majority of filtrate is reabsorbed in good. So option B is correct. The proximal convoluted tubule is where we have the majority of filtrate reabsorbed. Remember, we have our nephron, we have our proximal convoluted tubule and our loop of Henle, and then our distal convoluted tubule and our collecting duct. Once we get filtration, which is through that glomerulus, so this is filtration, reabsorption happens where it goes back into the blood, and then we get secretion happening to the distal convoluted tubule and collecting duct. This is secretion, reabsorption. Okay, so kind of try to remember this drawing here. Okay, question 17. Secretion happens mainly, I kind of gave this one away, so hopefully this one's quick for you guys. Which of those? D, right? So the distal convoluted tubule is our main site for secretion. Good. All right, question 18. The countercurrent multiplier system. Which of these is true about the countercurrent multiplier system? Good. So our answer would be E or none, because all of those are true. Sorry, all of those are false. Sorry, I said the opposite of what I meant to say. So it regulates blood pressure, not heart rate. I saw Elena, you said heart rate. I know you were thinking blood pressure. It doesn't regulate acid-base balance. It would be regulating water-salt balance. Remember that we have our loop of Henle, and then we have the blood flows the opposite direction. So salt comes into the blood first. I'm going to put an S. And then water is going to follow. I'll put a water. So the descending limb of the loop of Henle and the ascending limb of the loop of Henle. So out of the ascending limb, salt leaves. out of the descending limb water leaves. So the way that you could remember this, ascending, salt, S in ascending for salt, and then descending, like SAAS, salt. Descending is going to be for water. Okay? I'm just refreshing your memory on that. Okay, question 19. ACE enzyme true. Which of these is true about the ACE enzyme? Good. So the ACE enzyme is angiotensin converting enzyme. It's produced by the lungs, angiotensin converting enzyme. It is made by the lungs and it converts angiotensin 1 into angiotensin 2, right? So it's responsible for converting that angiotensin 1 into angiotensin 2. All right, question 20. Aldosterone is which of these? Good. So aldosterone is a hormone and it is released by the cortex of the adrenal gland. Released by the cortex of the adrenal gland. Okay. So aldosterone is that hormone released by the cortex of the adrenal gland. It is triggered to be released by angiotensin 2. So remember, angiotensin 2 gets converted in the lungs and then it goes to that cortex of the adrenal gland, triggering aldosterone to be released. Question 21. The macula densa are specialized cells. Which is not true of them? All right, so we know that they do line the bottom of the distal convoluted tubule. They are a part of the juxtaglomerular apparatus. They do release renin, but they do not release aldosterone. Remember that the aldosterone is released by the cortex of the adrenal gland, not by the macula densa cells. Okay, so if you answer D, that is the correct answer. Good. You guys seem like from your guys' responses, it seems like maybe kidney is a better section for you. So just kind of based, I kind of did this last time, just based off of how we did on this first part, I would recommend you guys to go over senses again. It seemed like, I don't remember, and I'm not calling anybody out. I don't remember who was answering what. I'm just telling you that overall, it seemed like you guys were struggling to get these questions right on senses. So I would recommend that you guys go over your senses chapter again. It seems like maybe that's a weak point versus the kidney. You guys are doing a lot better on those. So this is an area where like study it, but maybe you need to go back and study the senses again. Okay, question 22, the juxtaglomerular apparatus. What does the juxtaglomerular apparatus do? Good. So we know that the end result of the juxtaglomerular apparatus, which is the proximal, which is the afferent arterial, and macula densa, that system is also called the renin-angiotensin-aldosterone system. And this system is activated when blood pressure drops. Okay. And so the end result or the end goal of the system is A, to raise the blood pressure. So the goal of the system is to raise the blood pressure. The goal of the countercurrent multiplier system is also to raise the blood pressure. And the goal of the antidiuretic hormone is also to raise the blood pressure. So all of these systems in your kidney want to raise the blood pressure. The heart rate is governed by the brain. The kidneys help with blood pressure. Did everybody hear that? So raising and lowering heart rate is done by the brain versus the kidneys are going to raise blood pressure. The kidneys do not really lower blood pressure. Good? Everybody with me? So lowering blood pressure isn't really something your body does a whole lot. Okay, so that's kind of a system that lowering blood pressure, your body doesn't really care as much if your blood pressure goes high. It cares more if your blood pressure goes low. So the goal of the juxtaglomerular apparatus, again, to raise blood pressure. Question 23, which of these is not a component of urine? So we know that calcium is excreted in urine. Urea is part of urine, which is ammonia. Urea is ammonia and carbon dioxide, and creatine is also part of urine. So the answer here would be E, none, because all of those are components of urine. Question 24, kidney stones, which is not true about the cause of kidney stones? Good. So kidney stones are caused by the crystallization of anything. So all of those would be correct answers. So that's why the answer is none, because all of them are correct. All of them are true. All right, question 25. Which testy is lower, the left or the right? Good. The left, right? The left testy is lower. 26. Males have 23 homologous chromosomes. Good. So they have 22 homologous chromosomes, right? Just like females. In males, the 23rd pair of chromosomes is X, Y, whereas in females, the 23rd pair is XX. So females have 23 homologous chromosomes, females. Females. Males have only 22 homologous chromosomes, and the 23rd pair is an XY, which we call a heterologous chromosome. Heterologous. Okay? Does that make sense? All right, so moving on to question 27. The cremaster muscle, which is not true about it? So remember that the dartus muscle is the one that lines the scrotum. So the answer to this question would be D, because the cremaster muscle is made of smooth muscle. It is controlled by the autonomic nervous system, and it is sensitive to temperature. Its function is to basically lift the testes or the scrotum from the floor of the scrotum. So the way that I kind of think of this is like, if you know what a puppet master is, a puppet master kind of like, holds the puppet by the string. Does that kind of make sense? You could think of the cremaster muscle holding the scrotum by a string and then pulling it up. Hopefully that kind of helps you remember it. And then dartus muscle, you could remember it as it lines the scrotum to maybe dart from side to side. I'm just kind of trying to help you guys remember. So dartus muscle, dart from side to side. It lines the entire scrotum to help them pull up and inward, versus the cremaster is just going basically perpendicular, kind of like a string pulling the scrotum up. Hopefully that's a way to help you guys kind of make that stick. Okay, question number 28. The purpose of the inguinal canal is to allow the vas deferens to go up into the abdominal cavity. Therefore, it is not present in females. Is this true or false? True, right? So that is a perfectly true statement. It's just kind of a reminder to you guys what is the purpose there, and that is 100% accurate. That would be the purpose of the inguinal canal. Question 29. The male urethra is divided into how many parts? Good. 3 parts. Remember, it's the prostatic, membranous, and then spongy or cavernous. Which is not an accessory gland in males? Good. So we know that the seminal vesicle, prostate gland, Cowper's gland are all accessory gland. The Cowper's gland is also called the bulbourethral gland. The Bartholin's gland is also called the greater vestibular gland, and it's in females for lubrication. So we know that D would not be an accessory gland in males. It's in females for lubrication. Okay? So the answer there is D. Good job. All right, question number 31. Which cells produce testosterone? Good. The cells of Leydig, right? We can also call those interstitial cells. Cells of Leydig or interstitial cells. Either would be correct. Question 32. The pudendum is also called the Good. You guys know it's also called the vulva. The vestibule is the opening to the vagina. So like the entrance or opening to the vagina is the vestibule. Okay. Few more on this set and then, oh, that's it. Okay. So that was that set we finished. And then I have, let's kind of do some of these questions. Maybe let's do the nervous system because I feel like that might be a weak area for us. So let's do these nervous system questions here. Well, there's quite a few of them. Will you guys put in the chat for me if you have done all of these questions? Have you guys done all of these questions? Yes or no? Yes, no. No. Okay, if you've done these questions, have you also done these questions? Elena, have you done these ones as well? Looks like there's 70 in this one. Okay. Well, let's do these ones first because I'd rather do ones that no one's done. I'm assuming, we're all good on doing these ones, right? I think I think most of you haven't done these, right? So these ones are going to be a combination. It looks like I have, sorry, give me 1 second. Let me see. In terms of nervous system questions, it looks like these are all nervous system questions except for the first 15. So 70 questions and the first 15 are on muscle and then 16 through 70 are all on nervous. Okay, so let's do the muscle questions because on Monday you guys were kind of weak in the muscle area. And then let's kind of see how you guys do. And then we'll keep going and then start the nervous section. Just to remind everybody, all of these questions are available to you guys in the study resources module. So that way, if you're kind of like nervous on like, okay, well, what if I can't find these questions? Like, you don't need to try to write everything down. It's all available to you, but I feel like it is helpful to go through them together because we get more out of kind of discussing. Okay, so let's get started. Question number 1. Which of these, the characteristic by which a myofiber shortens is which? Good. Contractility, right? Elasticity, ability to return to the original shape. Extensibility is stretch. Question 2, for muscle contraction, which structure stores calcium? Good. E, the sarcoplasmic reticulum, right? The terminal cisterna will release calcium like gates versus the sarcoplasmic reticulum is the one actually storing the calcium. Question 3. Which of these does shorten during contraction? Does shorten during contraction. Good. So remember that the Z line or the Z disc stays the same. The M line is just the part down the middle. The A band is the length of myosin. It does not shorten or change. So we know that that H zone, which is the absence of actin, is the one that does shorten. So the absence of actin does shorten basically when the actin moves towards the center and pulls along the myosin, that will cause it to shorten. All right, question 4. Which of these does not shorten during contraction? Good. The A band, right? So the A band is our one that I just said does not shorten. It's where myosin is found and it's not shortening. The sarcomere does shorten, right? Because it's the entire contractile unit. I was worried I had a typo right there, but that is correct. Okay, question number 5. What substance travels in the T-tubule? Good. Sodium ions, right? So sodium ions travel in the T-tubule. Very good. Question number 6. Tropomyosin is exposed because calcium binds to which of these? Okay, so we know that our answer we're looking for is troponin. And since you haven't seen troponin, troponin is why we would choose E. So calcium binds to troponin or troponin. Question 7. Which of these does not need an ATP in muscle? A, right? Forming the cross bridge. So you do not need an ATP to form the cross bridge. Okay, it's breaking the cross bridge, recocking the myosin heads, and cycling calcium back that requires ATP, not forming the cross bridge itself. Question 8. Actin and myosin.
One really quick.
Yeah, go ahead.
We need to know the order, like the order in which the 3 ATPs are used, because when I was listening to re-listening to the lecture and then I re-listened to part of the SI and you and Knudsen said it differently. So I don't know. I just want to make sure that we don't need to know the order in which they're used.
No, you don't need to know the order. I mean, this should be the order right here. Breaking the cross bridge should happen first. Then cycling the calcium and recocking the myosin heads. Those are kind of like separate. Like it doesn't, one of those doesn't need to happen first. So in terms of the order, maybe you would want to know breaking the cross bridge would happen first. You know, that's kind of obvious. But that's all you would want to know. I wouldn't worry about the order there.
Okay. Thank you.
Yeah, no problem. As long as you know the three, you should be good on that. Okay, question 8. Actin and myosin, which is not true? Good. You guys got it this time, right? So they do not run the entire length of the muscle. Remember that myofibrils do, and the myofiber does, but the myofilaments, which are actin and myosin, they help make up the A band and the I band, and they are the thin filament actin, the thick filament myosin. They do not run the entire length of the muscle. Good. So A is correct. Question 9. What breaks open the synaptic vesicles? Good. Calcium. Very good. So calcium enters into that synaptic knob from the extracellular environment, breaks open those synaptic vesicles so that the neurotransmitter is released. Question 10. The muscle membrane portion that meets the synaptic knob in a neuromuscular junction is called which? And I'll give you a little hint for what I think this question is asking. I'm going to put the word specific, the specific muscle membrane portion that meets the synaptic knob. Good. So a sarcolemma is technically the same as the muscle membrane, but the real portion that we have that's part of the neuromuscular junction is the motor end plate. So the neuromuscular junction in textbooks and for your test is not the synaptic knob and the sarcolemma. It's the synaptic knob and the motor end plate. Does that make sense? So we're very specific about the neuromuscular junction being between the synaptic knob and the motor end plate. The motor end plate is technically a part of the sarcolemma, but it's the special part of the sarcolemma for the neuromuscular junction. So those two are not the same thing. So hopefully that makes sense. That's clear. Motor end plate is the specific wavy portion, you know, that little wavy part that synapses with the synaptic knob. So that's the motor end plate right there. Question 11, which does not belong for isotonic contraction? Good. So the way I remember it is you can pick up a gin and tonic, right? A gin and tonic is a drink. It's really easy to pick up. It's lightweight. So eccentric is you're reaching out for the gin and tonic. Concentric, you're pulling it back to you. Either way, concentric and eccentric, again, those are both examples of an isotonic contraction, just so everybody has that. Concentric, you're pulling it back. Eccentric, you're reaching out. Both are isotonic. And those are both force greater than load, right? It's easy for you to pick up a gin and tonic. Load greater than force is isometric. I've heard some students remember isometric as like a meter stick is flat. So when you go to the gym and you try to pick up 300-pound dumbbells, you're not going to be able to successfully pick up 300-pound dumbbells. At least I don't think anybody in here will. So you try to pick up that 300-pound dumbbell and it just stays there. That's isometric. Your load is greater than your force. Does that make sense? Question 12. When muscle cells increase in number, which of these is that? Good. Hyperplasia, right? Hypertrophy is an increase in size. Hyperplasia is an increase in number. So the way I remember that is you could think of like a trophy, like a big muscular guy holding a trophy. He's got big biceps. Does that make sense? Trophy is for size. Plasia is for number. Okay. So, I don't know, maybe you could think of like a place. A place has a lot of people. Number, plasia, large number. Trophy, I always remember like a guy holding a trophy, big in size. Question 13, cancer is which of these? Hyperplasia, right? So cancer is out of control cell growth. I'm not sure. Maybe the answer here should be neoplasia and not hyperplasia. Let's check 13 because I may have technically cancer is neoplasia. So it's possible for 13 we went with E. Yeah. So neoplasia is technically cancer. It would be out of control hyperplasia. That's why hyper is not correct. Hyperplasia happens like if we go to the gym and we work out. Hyperplasia is just getting more muscle cells, getting more muscle. Does that kind of make sense? Cancer isn't just hyperplasia. That's why I was wrong to say it was hyperplasia. Cancer is neoplasia. So it's when the growth never ever stops, and you get this tumor that's massive because it kept on growing. So you have to use neoplasia for cancer. So that's why E, neoplasia would be correct. Question 14, the agonist for flexion. The agonist for flexion. Good. It's the biceps, right? The triceps would be the antagonist, and the brachialis would be the synergist. Remember, the synergist helps the agonist. The agonist performs the action, also called the prime mover. The antagonist opposes it, and the synergist helps it. In case you're panicking and you're like, oh no, I don't know all the actions, for all these muscles. This is the only one you need to know, okay? So this is the only example that we give you to know biceps, triceps, and brachialis, okay? So don't be panicking, this is like an example, meaning we say, okay, what is the agonist for extension? The agonist for extension is the triceps, because remember, if you flip the movement, we would flip everything. So the antagonist for extension is the biceps, and the synergist for extension would just be another part of the triceps, right? Because there's like 3 heads to the triceps. Does that kind of make sense? So this is the only area we're asking you to know this example, and that's just so that we can kind of test that. I'm not saying 100% that I know it's on your test, but this is the only example we go over. Good. You shouldn't need to worry about coracobrachialis. Make sense? So we're not going to be pulling out random muscles all over the test and you don't know them. That's probably the only one we would use because, again, it's the only one we went over in lecture. All right, last muscle question, and then we're going to move on to nervous system. So smooth muscle is not which of these? Good. Smooth muscle is not striated, right? Smooth muscle has a fusiform shape. It's involuntary and autonomic, but it's not striated. Okay, so let's move on to nervous. So just kind of a little reminder going through, I'm just going to look at this one for you guys. So today we've done a good amount of muscle practice questions. We did quite a lot on Monday as well. We did a lot of kidney practice questions on Monday. Reproductive system, I also covered this on Monday. So today, with the last 30 minutes we have, we're going to go over nervous system. I did nervous system some on Monday as well. I just want to kind of make sure I'm really clear. The nervous system is a really long area of your text. You know, first you have the nervous tissue lecture, then you have the brain, then you have the spinal cord and the autonomic. So that's 4 areas or topics, and then the senses. The senses technically are part of the brain as well. We already did senses today. So you have 5 of the areas right there just on the nervous system. So in terms of my advice of like what you should study, muscle section, you need to study everything because really everything is going to be on that final. There's 160 questions. Muscle section, if you guys are feeling pretty good on it, I would kind of give it a rest. Focus more efforts onto the nervous section of your notes. Kidney, again, seems like you guys are doing pretty good on it. Again, I can't speak for everybody, but I'm saying kidney seems okay. Maybe give kidney a rest, focus on nervous. And it seems like you guys were doing good on reproductive too. So my advice to you would be to kind of hone in on some of your nervous area, your nervous system. Reminding you though, don't get too detailed into, you know, super detailed. You want to make sure you know all the basics really well. You guys in the past of questions we've done haven't really been doing as good on the basics, which makes me think, okay, maybe you're focusing too much on details. So make sure you have all the groundwork and all the basics down. And don't stress like so much on super extreme detail, but make sure you have every basic definition down. You know, what are the 3 parts of the diencephalon? What are the main functions of the 3 parts of the diencephalon? Okay, what are the 3 parts of the brainstem? What are the main functions of the 3 parts of the brainstem? Those are things you need to be saying that and knowing those really quickly. If you're kind of struggling and you're like, well, you know, I studied, you know, this part for 20 minutes, but I don't really remember the parts. You need to like be looking at make sure you have those main bullet points really nailed down, because when you get to the test, you're going to want to know all that main stuff. Does that make sense? Make sure you look at the detail because it could be on there, but I'm just trying to encourage you, like, don't get so lost in those details that you miss the main key points. What are the 3 parts of the diencephalon? What do the 3 parts of the diencephalon do? Are you so lost in detail that you kind of forgot to go over the main stuff? So I'm just trying to help direct you. Make sure you do go over the detail as well, because that is stuff that will be on the test, but make sure you also get all those key points. I recommend making your own outline of the different lectures, especially in the nervous section, because it will help you. Okay, let's start with question 16. Nervous system by structure. Which is not true of the nervous system by structure? So the answer here should be none, because the nervous system by structure constitutes the brain and the spinal cord and everything that comes off of it. Nervous system by function is afferent, efferent, etc. So let me go check just to make sure 16 is E. Sometimes I like to double check so I'm not accidentally saying the right. Yeah, so E, all are a part of, all are a part of the structure. All right, question 17. The functional nervous system or afferent nervous system, which of these is true? Good. So our answer here is B. This is kind of a tricky question. Remember, afferent is the same as sensory. Okay. So it would control autonomic sensation of the sensory, and it would control somatic sensation. But remember, somatic is what you are aware of. So are you guys aware right now of your cardiac muscle pumping your heart? No, right? Are you aware of the hormones being released from your body? Glands? No. So somatic would be things like skeletal muscle, skin. Does that make sense? So somatic sensation is the afferent system, but not of smooth muscle, cardiac muscle, and glands. So B is correct because it is the autonomic or visceral or subconscious sensation of intestines, heart, and lungs. Hopefully that makes sense. If you guys have questions, as always, just please feel free to unmute or put it in the chat. Question 18, neurons, which is not true about neurons. Good. So neurons don't regenerate. Neurons are maintained by glial cells, but they do not have the ability to regenerate or perform mitosis, right? So neurons do not consistently regenerate. Question 19. Which is not always present in a neuron? Okay, so remember, an axon is not always present. We have some neurons that are called anaxonic, which means no axon. So it is possible to have a neuron that does not have an axon present. So that's called an anaxonic neuron. So the answer there would be C. Question 20, the unipolar neuron, which is not true about the unipolar neuron. Good. We know it's found in the dorsal root ganglion. The dorsal root ganglion is also called the spinal root ganglion. Ganglia is the plural form of ganglion. So spinal root ganglia is the location for unipolar neuron. It's kept alive by satellite glial cells, and it's located in the PNS. It is not found in the anterior spinal cord. It would be found in the dorsal or posterior spinal cord. Good job. Question 21. Which cells make up the blood-brain barrier or choroid plexus? Good. Astrocytes and ependymal cells, right? Together, those two make up the blood-brain barrier or the choroid plexus. Question 21. The blood-brain barrier or the choroid plexus creates So remember, what does it filter, right? It filters blood to create cerebrospinal fluid, which is purified blood. So it doesn't filter lymphatic fluid, right? It's filtering blood. So the answer here, I believe, is E, because it does not create filtered lymphatic fluid. It does not remove all alcohol and poisons. Remember that, you know, if you have a baby, if a woman is pregnant with a baby, the blood-brain barrier of the baby's brain cannot filter alcohol, all alcohol and poisons. That's why you can, you know, you don't drink alcohol when you're pregnant, because the blood-brain barrier does not filter alcohol. That's why when you get drunk, alcohol is intoxicating or getting into the cerebrospinal fluid. Does that make sense? So it does not filter all alcohol and poisons. It should not be a filter for lymphatic fluid. It's a filter for blood. It does not cushion neurons. That's not the point of it. It lines the ventricles of the brain, not the lobes of the cerebrum. So the answer there should be E. Whenever it's E, I always just check to make sure. 22E. So it creates cerebrospinal fluid, which is purified blood. So it's not, the function there isn't to filter lymphatic fluid. It's possible some lymphatic fluid is getting filtered, but that's not like the main function of it. The brain has 4 regions. Is this true or false? You guys are making me second guess myself. Let's see. I used to always get confused on this. Yeah, so 4 regions of the brain. So I double checked my notes just to make sure. So the answer here is true. Okay, I know that we all said false in the chat. So the 4 regions are the cerebrum, the diencephalon, the brain stem, and the cerebellum. Those are the 4 regions. If you added the limbic system, that's not technically a region of the brain. Yes, so when I was in the class, I used to do this too. The cerebrum has 5 lobes. So when I was in the class, I kept doing the same thing. I kept thinking the brain had 5 regions because the cerebrum has 5 lobes. So don't make the same mistake I did when I was taking this. So cerebrum is going to have your frontal, temporal, parietal, occipital, and insula. But the brain only has these 4 regions. Good? Diencephalon, we know, has the thalamus, the epi, hypo, and thalamus, and brainstem has the medulla, the midbrain, and the pons, and then the cerebellum has only the cerebellum. Okay, question 24. A funiculus is not Okay, so we know that the funiculus is a large group of axons, which is also called a large tract when it's in the CNS. Okay. What is a large group or network of axons in the PNS? Can anybody do this fill in the blank style? So we know a bundle of axons in the PNS is a nerve. A bundle of axons in the CNS is a tract, right? So you have your axons. Oh no, I'm going to just try to do this quickly. But this is something that you guys want to remember. So a bundle of axons in the PNS, nerve, in the CNS, tract. But what about when we get a really large tract? So I'm going to put a L. Oh, that was supposed to be an L. Okay, wait, L. And then a large nerve. So a large nerve network is called a nerve plexus. A large tract is called the funiculus. Okay, and we're about to do the next part, which is going to be cell bodies. Okay, so let's answer question 25, and then I'll kind of draw the little diagram over on the left. So a center is which of these? So let's answer question 25. What is a center? Okay, so first we have clusters of cell bodies. And if we have clusters of cell bodies in the peripheral nervous system, it's called a ganglion. If we have clusters of cell bodies in the central nervous system, it's called a nuclei. Now, when we join a lot, I'm going to draw like, maybe that will work good, like a big arrow. When we get a lot of nuclei together or a lot of ganglion together, there's different terms. I'm going to show you this. So a lot of nuclei together is called a center. This serves a specialized function. So what that means is like you could have a center for vision or a center for hearing. It's like all these nuclei get together with the same function. And I did a little tricky thing over here. There is no equivalent, no equal. So I'll write a big X. So there's no bigger form of the ganglion. Does that make sense? So the ganglion is just the ganglion. There's no bigger form. The nuclei get together and become a center. So looking at what my diagram says, let's re-answer question 25 and see if we can get to the right answer. What would the correct answer to question 25 be? Okay, so clusters of cell bodies is going to be the nuclei. But a center, right, is large bunches of nuclei in the CNS. So it's like you get enough nuclei together, you get a center. Does that make sense? Okay, you guys are getting it now. Okay, good. Okay, let's answer question 26 to hopefully get this concept. Question 26. A ganglion is which of these? So remember, a ganglion is where there's a synapse between a cell body in the peripheral nervous system, right? So we have our original one in the anterior horn, and it goes out and it synapses with the next one, and we called that the pre-ganglionic neuron and the post-ganglionic neuron. And we know that we studied those as part of the autonomic nervous system, right? So a ganglion is this part right here. It's basically where there's this cluster of cell bodies in the peripheral nervous system. And the function of ganglions is actually to be a part of the autonomic nervous system. So answer A would be correct because the ganglion is part of the autonomic nervous system. The ganglion is the cluster of cell bodies in the peripheral nervous system, right? Cluster of cell bodies, peripheral nervous system, and it is part of the autonomic nervous system. You would have the pre-ganglionic neuron and the post-ganglionic neuron. And remember, you guys need to know where they synapse. Remember, we said terminal ganglia, synapse is near the wall, intramural is in the wall. Then you had the ones that synapse in the sympathetic chain of ganglia or near the sympathetic chain of ganglia. Is that kind of ringing some bells? You had your sympathetic system, parasympathetic system. So you kind of want to take another look at that for the lecture. So, you know, what would you want to know for the autonomic lecture? Your main thing you want to know is length of the pre and post ganglionic. So like, is it long? Is it short for sympathetic, parasympathetic? You want to know the location of the synapse or the ganglion. And so that would be, you know, terminal, intramural, all of that. And then the other names for the systems. So like sympathetic is also called thoracolumbar, and parasympathetic is also called craniosacral. And then you want to know their basic functions. So that should be most of the main topics. I do want you guys to not see that as like the only answer key for what you need to know on the test. I don't know all that you need to know. That's a long chapter. I'm just helping you guys to kind of narrow it down to some of the key points. So those are some of the key points you would want to take a look at. I would recommend if you guys have a recording of my SI session on that, and you guys can just fast forward to the part of the recording where you find my outline, try to find my outline and make your own outline from it. If you kind of want, like, honestly, an advice how I would study if I was studying for the final and I had access to all of the SI recordings, I would fast forward the SI recording to the part of the SI recording where I just have my outline pulled up. And then I would just pause it on that screen and rewrite the outline on my notes. If you want, like, kind of what I would do, I wouldn't rewatch all the SI sessions. I would just scroll over whatever buffer over until you find the part that has the outline and rewrite your outline, kind of using mine as a guide if you want my suggestion of how to study. Because then you guys are going to review all of your notes, which is the most important thing. Is that somehow you're reviewing everything in an active way. So don't just sit there and read your notes to yourself. You need to actively engage. One of the best ways to do that is making outlines. So question 27, CSF does not flow through which of these? So this is a very tricky little question that I came up with here. So the septum pellucidum divides the ventricles, the lateral ventricles specifically. So the answer is actually D, the septum pellucidum. The sinuses, when you read this, you were probably thinking of like your sinuses, like when we all say my sinuses are congested, we're talking about like our nasal sinuses. There are sinuses, really, you know, the best way to write this would have been sinuses in the brain, but there are sinuses in the brain and these reabsorb used CSF. Do you guys remember that? They have arachnoid villi sticking into them with arachnoid villi sticking into them. Is that kind of ringing a bell? So those sinuses do reabsorb use CSF. So CSF does flow through them. I know you guys are probably thinking of like your nasal sinuses, but these are different. They are the ones that reabsorb use CSF. They have arachnoid villi. Septum pellucidum, again, is that structure that is dividing the lateral ventricles. So you're going to choose D because that would not be allowing CSF to flow. Hopefully that kind of makes sense there. All right, question 28. The arachnoid villi are found where? They are found in the sinuses, right? Maybe you guys weren't answering because I already gave you the answer. So arachnoid villi are found in the sinuses. They help to reabsorb used CSF. Okay. The sinuses are basically a part of the subarachnoid space. But subarachnoid space wasn't there and sinuses was. So that's why we chose sinuses. Okay, question 29. The gray matter. What does the gray matter not have? Good. Because we know white matter is myelinated axons, so gray matter does not have myelinated axons. White matter has myelinated axons, so gray matter does not have myelinated axons. Good. Question 30. The cerebral hemispheres are divided by This is a very strange question. Let me see. I'm looking at it and you guys are answering longitudinal fissure, which seems right. I'm gonna go with, let's look at the answer. That one's kind of tricky. I don't know. I'm not actually 100% sure what the answer is there. I think it should be A, 30A. I agree with you guys. You guys are answering A. 30A. Yes. Longitudinal, where am I at? Longitudinal fissure. For some reason I was looking for I'm like losing my brain cells. I was looking for the largest tract in the brain, corpus callosum, but corpus callosum does not divide the hemispheres. So you guys were right. The hemispheres of the brain, the two halves, would be divided by longitudinal fissure. Okay, question 31, the primary somatosensory cortex, which is not true about the primary somatosensory cortex. Okay, so we know that the answer here would be D. So it is not located rostral. Remember, you can think rostral is nostril. So it's not rostral or nostril closer to the nostrils. related to the central sulcus. It would be posterior from the primary somatosensory cortex. Does that make sense? So the answer there is D. Question 32, the primary olfactory cortex is found where? Temporal, right? So the sense of smell goes to the temporal lobe. Good. Question 33. Wernicke's area is not. This is a tricky question. Let me make sure I've got that. Yep. So you guys are so Wernicke's area is not for writing. So remember you have Broca's area for speaking and writing. You could think of it as like the action area, like what you do for action. You act and you speak, you act and you write. So that's why it's in like the frontal lobe for action versus Wernicke's is more like you listen, you understand what's written, you understand what's spoken. So that's why D is correct. Okay, question 34. Cerebral nuclei. We just talked about nuclei, right? So what are cerebral nuclei? So we know that cerebral nuclei are not part of, I'm going to try to eliminate the easy ones. We know they're not part of the PNS. We know that they would be CNS because nuclei are part of the CNS. So it can't be D. They don't involve the cortex of the cerebrum. So the cortex of the cerebrum is, if you think about the brain, the cortex is the outside. And then we have the brainstem. Sorry, I know this is kind of like a crude drawing of the brain, but we have the brainstem like at the center, right? This is just to kind of help us kind of orient ourselves. And then we have our white matter that basically is all of this part in the middle. This is white matter. So it's not part of the white matter because they're nuclei, and we know white matter are axons. So they are deep regions of gray matter. So the cerebral nuclei are little clusters of gray matter kind of located around the cell, the brainstem right here. So our answer would be A, they're deep regions of gray matter located around the brainstem. The cortex of the cerebrum has nuclei as well, but they're not the cerebral nuclei. The cerebral nuclei are the ones located around the brainstem. Okay? All right, question 35. The diencephalon does not So we know that it does not help control respirations, right? Because the diencephalon has the epithalamus, which is for circadian rhythm, has the thalamus, which is a switching station. The epithalamus has the pineal gland, which produces melatonin. That's why it regulates circadian rhythm. Then it has the hypothalamus, which produces the hormones and sends them to the pituitary gland for storage via the infundibulum, right? Via the infundibulum, which is like the tube that connects them, infundibulum. And then you have our brainstem. And the way I was kind of telling you guys to remember it, brainstem, M, think midbrain. Midbrain doesn't really have a function, honestly. It's kind of just like a connection point. Pons, you could think breathing, B for breathing. So that's the pons. And then the medulla oblongata, which has the pyramids for decussation or crossing over of the white tracts. And then it has as well a cardiac center for heart rate, a vasomotor center for blood pressure. And then finally, it has a respiratory center for things like coughing, gagging, sneezing. So I know that was obviously long, but that's just a recap of those two areas. And that's top one is the diencephalon. Okay, let's answer a few more. We got started, 10 minutes after 9, so I'm going to go a few more minutes here. So question 36, Schwann cells, which are true about Schwann cells? So the oligodendrocyte is not a Schwann cell, right? The oligodendrocyte is the glial cell in the CNS. It's not a neuron, it's a glial cell. It doesn't produce myelin in the CNS, it produces myelin in the PNS. And it does help to speed up the rate of depolarization by producing myelin, but technically the Schwann cell is not the one doing that. So I would answer E. So I would answer E again. It's not speeding up the rate of depolarization itself. It's producing the myelin that speeds up the rate of depolarization. So E is what I would choose. Okay, question 37. The infundibulum. The infundibulum, what is the answer there for the infundibulum? Good. You all got it. Connects the hypothalamus to the pituitary gland. Question 38. The superior cerebellar peduncle. Good. B, right? So that's the superior. The middle, remember the peduncle is just a connection for the cerebellum. The middle cerebellar peduncle connects the pons to the cerebellum, and then the inferior cerebellar peduncle connects the medulla to the cerebellum. So that's the inferior cerebellar peduncle connecting the medulla to the cerebellum. The vasomotor center, which is not true about the vasomotor center. Good. C, right? So we know the vasomotor center acts on smooth muscle. We know it regulates blood pressure, and we know it's located in the medulla oblongata, but it is not located in the hypothalamus because the hypothalamus is for producing hormones. Question 40. The pons mainly functions for good. Respiratory depth and volume, right? So we know the pons is a respiratory center. That means it's specializing in respirations. It's helping in respiratory depth and volume. Okay. The medulla also has a respiratory center. Medulla oblongata also has a respiratory center, but this is like a minor respiratory center, and it basically is like a helper. So this is things like coughing, gagging, sneezing, vomiting. Is this making sense to everybody? Actual respirations, like what makes you breathe in and out, comes from the pons. So if a person got in a car accident and their brainstem is damaged, this is why usually in the hospital they'll be on a ventilator because they cannot breathe. You've heard of a person being on a ventilator after like a car accident, being in a coma. That usually happens when the brainstem is damaged because their pons can no longer help them respirate or breathe, and they have to be put on an artificial ventilator to breathe for them. Does that make sense? So the pons governs respirations. The medulla helps. The medulla is also in the brainstem. It helps, but the respirations mainly, the main function of the pons is for respiration versus the medulla, it's like another function, right? Because the medulla also has the cardiac center for heart rate and the vasomotor center for blood pressure, and it is the switching station via the pyramid. So the medulla has like 4 different functions versus the pons only does respiratory. Okay, you guys look like you're still with me. We'll do a few more. Question 41. I want to get to the end of the brain questions here if possible. The cerebellum is not for which of these? Good. We know it's for fine motor control, not coarse motor control. Okay. So fine motor control is the cerebellum. It functions for proprioception or knowing where your limbs are. and it helps with equilibrium or posture. Okay. Are you guys good with going to question 45? It's only like 4 more questions, and then that kind of takes us to about the 2-hour mark. So question 42, which of these does not belong for the limbic system? So the answer here would be E. All of those do belong. All of those are part of the limbic system. The limbic system has the amygdaloid body, the hippocampus, the cingulate gyrus, the caudate. Oh, never mind. You guys answered D, I think. D should be right. Let's go check the answer. I like to do this so that way, in case I'm saying it wrong. So 42D. This is honestly a way too hard of a question. It's not going to be that hard on the test, but this is a more challenging question than you'd get on the test. But A, B, and C are all part of the limbic system, plus the claustrum and the parahippocampal gyrus. The caudate nucleus is for walking. And this is not part of the limbic system. So technically D is the answer, but I'll just kind of tell you that the test won't be that complicated on that topic. Question 43, cranial nerve 7. What is the action of cranial nerve 7? So this is where I told you guys you need to go over these on the test. There will be cranial nerve. In Roman numeral, you need to know all the Roman numerals. You need to know the cranial nerve names for the number, right? And the basic function. I recommend that YouTube video that I showed you guys in the module. It gives the basic functions, okay? So on the test, I remember at one point I went back to the test to count. And there was, I think, 8 or 9 questions just on the cranial nerves. So I don't want you guys to be surprised or thrown off. That's 10 points potentially on just the cranial nerves. So study them, go over them, the Roman numeral and the function and the names. Okay. I believe cranial nerve 7 is, I think, the facial nerve. I think that it's C, but honestly, I kind of memorized those. Yeah, so 43C. I'm just telling you quickly because if you guys haven't studied those are just straight memorization. So it's not really, you can't really reason your way out of these. So again, I'm not going to go to 45 because now I see that these are just on them. I would just tell you guys, find a mnemonic for them. So I'm just going to end on this to remind you guys where this is. So if you go to your YouTube videos and helpful links in the module, right here, is it going to load for me? See if it loads. At the very bottom, there's a cranial nerve video. It has a mnemonic. They do like singing. It's only like 3 minutes long. It has really good basic functions, and it's really helpful. I watched it a lot when I was learning it. They go over the mnemonic at the very end to kind of help you guys remember them. So I found these very, very helpful. I would recommend this video for learning the cranial nerves. Again, it's on the exam along with everything else. Okay, any more questions for me before we go? Any more questions about the test? I believe Bender, if you guys are here, I know that at least 1 or 2 of you are from Bender's class. Bender might have a different number of questions for the final. You want to confirm that with him. Knutson has 165, maybe even 170 if you're taking Knutson's class. But Bender might only have 100. So, and he might even be doing 50 or 75 worth 2 points each. So If you don't know already and it's not already clear to you, send him a Canvas inbox message so you kind of know what to expect in terms of number of questions. I'm sure you guys already know, but just in case you don't. And yeah, thank you guys for coming all semester long. I really appreciate it, and hope everything goes well for you guys. And let me know if you have questions. You guys can Canvas inbox me about anything, including nursing stuff. And you should have the nursing recommendations link in your announcements section. This is kind of like my nursing recommendations. You guys can read through and save this document for later. I am here until noon. And if you guys don't have any other questions, I will let you guys go. Thank you guys again. Veronica, it depends on which teacher you take. I know that for one of the physiology classes, there was an SI at one point. I would imagine that, there's still an SI, but it just depends on what teacher you take. I know that I would tell you that the overall answer is no. Overall, no, there aren't other SI. I didn't run into a lot. I haven't known a lot. I know most of the SI leaders, but at times there have been physio SIs. Micro, I don't know. I think, actually, you know what? I did know one girl who was the SI for micro for a little while. I don't think she's doing it now. So iffy. I would say you have like a 50-50 chance. Anatomy has, because I do both Bender and Knudsen, anatomy, I've been doing this for a long time, so we have like the most SI, but I think I'm the only SI for anatomy. So, yeah, there should be SI for all classes because I feel like it's just so helpful. But unfortunately, it's not something that happens super often just because I think professors don't always push for it, like Knudsen pushes for it, and Bender, so then it just doesn't happen, unfortunately. Thank you. I'm really glad it helped you this semester.